Many of us are interested in specific metabolites, and we typically use simple biochemical assays or maybe even mass spectrometry-based technologies to assess the level of these metabolites. But many metabolites are present in different cell organelles. They are also maybe have different concentrations in these different cell organelles. And we don't have the technologies available to dissect where in the cell, for instance, the specific metabolites are affecting. We also typically use a piece of tissue or maybe a whole dish of cells for those assays. By doing that, we are actually confounding our results, either by analyzing cells that we are not really interested in, or maybe simply due to the lack of the spatial aspect. But researchers are pushing the boundaries. They're refining methodologies and technologies to actually do spatial metabolomics and to quantify metabolites in single cells or even in single organelles. And one of the pioneers in this field is our next speaker, Professor Ingela Langenkopf. Ingela has a background in analytical chemistry with a PhD from Gothenburg University. She has, since her postdoc work, which was conducted at the Pacific Northwest National Laboratory in the US, studied or tried to advance mass spec-based methods to quantify metabolites in spatial and using spatial and cellular metabolomics. And she's now a professor at Uppsala University in Sweden. So Ingela, we look very much forward to your talk with the title Spatial Metabolomics, Deciphering the Chemistry of Biology.
Great. Thank you so much. Thank you for the introduction, and thank you very much for the opportunity to talk to you guys here today. It's great to see so many people here. It's wonderful. And also all the people I've interacted with thus far have been really great. So very nice community here, I must say. Yes, so as you already mentioned in the introduction, let me see here. This is the typical way that we deal with cells or tissue when we want to analyze them metabolically. So we take them all and we homogenize them, put it into some sort of vial or other solvent, and then we do some sample cleanup to remove things we're not interested in, and then run the analysis with LC-MS. And this is a great tool, and I know lots of people that are doing great work with this, and we should not stop doing this at all. But it doesn't really give us information about where metabolites are localized in, for example, a brain tissue here, as we can see, that has many different morphological regions, cellular regions that are specialized in different activities in the brain. And it also doesn't really tell us about the variability between individual cells. So in my research and my research group, we are trying to work to develop techniques that can sort of complement the current LC-MS-based strategies. And one of the major techniques that we're using is called nanospray desorption electrospray ionization. And we're doing this for mass spectrometry imaging. This is what I will talk about today, but we're usually doping it with different analytes or other dopants to see what we can get. We also have this as a pneumatically assisted version. And in addition to this, what we're doing in my lab is that we're developing this for single cell metabolomics. This I will not talk about today. But we can do this, so then we can actually get the metabolome from individual cells, which is quite exciting. We're also doing something that we call surface sampling capillary electrophoresis mass spectrometry. And that is because with imaging mass spectrometry, we're lacking that dimension of separation before mass spectrometry. So we are working on some tools to get that part in as well. And the final one doesn't actually have to do with spatial metabolomics, but rather with analyzing few and precious cells. So with our tool that we call direct infusion probe, or DIP, we can actually use just a few hundred cells and get the metabolome of those. So you don't have to use a million cells like you normally would do for metabolomics, but we can scale this down. So that is sort of the concept of what I want to contribute with my research, is tools that we can actually go beyond what we can do today. But anyhow, what I will talk today about, as I mentioned, is mass spectrometry imaging, and in particular then nanodesi. So the data that I will show you, some of it is derived with using the DOPE nanodesi, but most of it is actually with the DOPE pneumatically assisted nanodesi. It's the exact same tool, with the exception that, let's see if I can do this now, let's figure out how it works. With the exception here that we actually have a nebulizer on the spray capillary. So I will tell you a bit about how this works, and I will show it all as the original tool. So what happens here with nanodesi is that we place it in front of the mass spectrometer. So how many people here are familiar with mass spectrometry? Oh, great. It's a majority, I think, but just for the rest of you, what we do with mass spectrometry is that we can detect molecules, but first they have to be ionized to become ions. And then we can detect ions based on their mass over charge. And I will talk today about singly charged ions. So it's basically the mass of the ion that we're talking about here. So with nanodesi, what we do is we place these two fused silica capillaries. They're quite small, so 150 micrometers in outer diameter and 50 micrometers in inner diameter. We place them on this angle directly in front of the mass spectrometer. So now we have stripped the mass spectrometer of everything that's LC or a conventional electrospray source or anything. It's just the bare mass spectrometer, and we put these capillaries in front. We can then run a solvent through the first capillary, and this solvent will create a little liquid bridge to the next capillary. And this can then move on to the mass spectrometer for electrospray ionization of our molecules and then detection in the mass spectrometer. Now the key here is this little liquid bridge that is formed between the capillaries, because as this liquid bridge touches on a place on the surface, molecules from the surface will be desorbed into the running liquid bridge and then directly transported into the mass spectrometer. So that means that we can get a profile of the molecules that are at this specific site of the tissue or whatever sample we have under there. So yeah, that's just a schematic mass spectrum. So on the x-axis here, they're mass to charge, and as I mentioned, we'll look at singly charged, so it will be the mass of the ion and then the intensity on the y-axis. So how do we then do imaging with this? Well, what we do is that we place these capillaries, we place a tissue section, just a tissue section like you would use for histology or something else, just a thinly sliced one, under these the liquid bridge, and then we basically move the tissue section under this liquid bridge while we're continuously acquiring data. So that means that we're continuously getting data from different positions on the tissue. And the data that we're actually getting looks something like this. So this is a mass spectrum, and you can see we have metabolites and we have phospholipids, and it looks kind of empty in the middle, but it's actually not. It's a very rich mass spectrum. And this is what we find in every pixel, so everywhere. But of course, it will shift depending on what we are detecting at the specific location. So if we just take a look at the phosphatidylcholine-rich region here, this is what it looks in the zoom in, and we can see if we're looking at this mass here at 20.5265. Yes, all the numbers are important and we can detect them because it's a high resolution instrument. We can see here the distribution of how this specific ion is distributed in the tissue. And we can see here that we have a color scale, so the darker is the less intensity, and the brighter is the highest intensity. And we're looking here at phosphatidylcholine PC36-4, which is also on this it's really hard with this one, molecule here. So we are summing up the number of carbons in the acyl chains to 36, and then the double bonds are 4. So we can take a look at another one like this at 826, and we can see this distribution. Now, as you can see, it's almost the opposite distribution. Even though they're very similar, the only difference is actually 3 double bonds. And we can move on and look at the 848 ion here. It's PC38-4. So compared to 36-4, it's 2 more carbons and a totally different distribution. And another one is like PC40 and 6 with 40 carbons, 6 double, yet another distribution. So I think this is quite interesting, and this is why I got into the field in the first place, because we can see so many differences that I don't know what they mean, to be honest. I don't know if anyone knows what they mean, but it's quite exciting. And then we can try to start to understand what they mean, because in each pixel of our mass of our ion here, or ion image here, we have a mass spectrum. So in each pixel, we'll have this unique fingerprint of the molecules that are in this specific pixel. But of course, there is nothing such good as everything works with instrument or techniques, right? So there are some really good big limitations with mass spectrometry imaging. And these are in sensitivity, because we are detecting all ions at one time, right? So you know how many molecules there are in tissue, and in one event we're going to ionize all of it, which means that low abundant or low ionizable molecules will not be detected so much. So sensitivity is an issue. The other issue is specificity. As I showed you, we're summing up the carbons of the acyl chains, the double bonds, to show this is a PC. That's not very specific because there are lots of isomers and isobars. So today, I will tell you how do we deal with this in my research group. And in particularly, I will tell you about silver cationization. So silver cationization we do together with nanodesa imaging by introducing silver ions in our solvent. So that the solvent that continuously drives through and desorbs analytes and then ionizes analytes. So what happens is we have the Dewar model that shows that silver interacts with double bonds, with the p bonds and the d orbitals. And in particularly then for mass spectrometry, this means that we will have a third category of the kind of ions that we detect. So normally in mass spectrometry imaging, we detect protonated, sodiated, and potassiated ions. But now if we add silver, we will also detect silver ions, or silver adducts, which is arginated ions, and they are much higher in intensity. So if we look at, this is a steroid molecule here, we can see in the red bars, we can see we have protonated ions, we have sodiated ions, and then we can see here silver as the 2 isotopes, 107 and 109, here much higher in abundance in for the steroid. And actually, if we look at several different types of steroids, we can see that this is a calibration curve, and we have on the bottom, we have the protonated, on the top, we have the arginated, and we can see that it's a much higher sensitivity for using the slope as a measure of sensitivity. It's in between 2 and 150 times increase the detectability of these steroids. Similarly, if we look at prostaglandins, which are typically visualized in as deprotonated, which are then the yellow and the green bars. If we instead look in positive ion mode as arginated, we see that we have about a 30 times increase of these. So sensitivity here with silver adaptation for some molecules, of course, not for everything, but it's quite significant. And so I will tell you some stories, and I will be a bit brief, especially in the middle here. in the interest of time, but about how we are then visualizing this. And the most important thing is that we are using silver adaptation together with tandem mass spectrometry. So with tandem mass spectrometry, if we have isomeric precursor ions, so ions with the same mass to charge, we can then add some energy, they will break up, and we get product ions. So we can then see what they are, what products they are made of. And we can select one of these further to, again, break it up, and we can see what the product ion is made of. And this is what we refer to as MSN. And one example of, I'm moving on. And one example is then the phosphatidylcholine species that I showed you previously here. If we don't know the position of the acyl change in the glyceryl backbone, we abbreviate it as 16-0-18-1 if we at least know about them, otherwise we sum them up. But what we would like to know is, of course, where they are situated. So can we then identify these when using silver and MSN? And of course we can. So if we look at MS1, we only see the precursor ion here. And moving forward to taking this into MS2, we can see how it breaks apart. We get a dioxylin ring formation. And moving forward from this, we're breaking that apart here. We get loss of silver hydrate. And moving forward again here in MS4, so breaking it down 4 times, we actually get some significant information about the acyl chain position. But we have to go all the way to MS4 to do this. So what that means is that we can we have confirmed this for several glycerolipids, but also for fully saturated. So it doesn't have to be unsaturated glycerolipids for this, and no need for high-res. And it works very well with imaging. So when we do imaging, then in addition to, in addition to silver ions, we put internal standards into our solvent. This is because then we can get a quantity of how much we're detecting. So if we're then looking at the PC34-1, as I said, this is the sum of the carbons in the acyl chains, we can see that it breaks down into 2 isomeric structures, SN positional. So we have 16-0-18-1 and 18-1-16-0. And if you look at these, they look extremely similar, right? So it looks like they're distributed in the same way. However, if we do a ratio image of this, divide one by the other, we see it's actually quite big differences where we have the 16-0-18-1 more in the white matter regions, but also in some subhippocampal structures. Another isomer here at PC36-1, we can find the isomers 18-0-18-1 and 18-1-18-0. Now, when we do the ratio image here, we can see they're distributed in the exact same location, which means that they're everywhere in the same ratio. But we can see another isomer pair here of the 21-16-0 and 16-0-21, all 4 hidden with below the 36-1. And when we do the ratio image here, we can see that there are also some differences in the distribution between these. We can take it one step further and look at regions of interest analysis. So in the regions of interest, we go to our data. We mark regions that we think are interesting, then we can extract information from those specific regions and compare with other regions. So here we have marked the cortex, corpus callosum, 2 regions in the hippocampus substructures. And then we can, since we have the internal standards, we can see how many micromoles that are detected in each of these. I specifically say detected, because it's what we detect. We don't know how much is in the tissue, because then we also have the extraction efficiency that we still don't know really how to deal with, actually. But here we have then, we can see that they're about similar, a little bit higher from the 16-0-18-1, but otherwise similar in the regions. If we're looking at the 18-0-18-1 and 18-1-18-0, these were distributed the same way, if you remember from the previous slide. But actually, when we look at it quantitatively, there's a big difference in how much is there. And then the 21, 16, 0, 16, 0, 21, we can see that they have a difference in the corpus callosum in particular, where the 16, 0, 21 all of a sudden becomes a dominant species over the other one. So this is quite interesting, but why is this so? I read some studies suggesting that there is a PLA activity difference between these morphological regions. I don't know if that's true, but I'm thinking, what about the membrane properties, right? So we have membrane proteins that sit in membranes, and somehow they must be interacting with these specific molecules, because otherwise, why would there be such a need for these very distinct isomers? So we have the plasma membrane, we have the ER, we have the Golgi. All of these are constantly dealing with this type of structure. So yeah, it's quite interesting to think about what this might mean. And if someone has any ideas, I'd be happy to discuss. But we also have double bond positional isomers, and I will very quickly just show you the trouble that we went into with this, looking at oleic acid and vexinic acid, which are then just different by the position of the double bond. So we have, again, using silver, and in the MS2, with one fragmentation, we see the loss of water and carbon monoxide, but in MS3, we get this. So what do we see when we look at these mass spectra? Well, what we see is that they're kind of complex fragmentation patterns. We see that we have the identical master charge of the product ions. So in the previous one, we had diagnostic product ions, but now they seem identical. So how do we deal with this then? Well, if we look closely, it seems to be different profiles, right? And so what if we then sort of titrate vexinic acid to oleic acid amounts and run all these standard samples? And then what we see is here, as we're changing the amount of the respective fatty acid, we do get a profile change. So we then went around, so what we did we developed a linear regression model for this, and here you can see the predicted ratio over the real ratio, showing that we don't have very much error in this prediction. And then, of course, we can image this as well. And this image showing then the brightest one is where we have the most oleic acid relative to vexinic acid, and the more red is more vexinic acid. If we put some numbers on this, we can see that oleic acid is overall more abundant, but vexinic acid is there at a slightly different ratio in the different regions. So with the double bond positions, of course, we know the omega-3, but I'm not going to get further into this here. But another thing of the prostaglandins, they are related from one of the fatty acids, right? So arachidonic acid via the COX-2. So this is an important, very bioactive molecule, and inflammation and vasodilation. So with the sensitivity that we accomplished with the silver, we have shown that we can image then prostaglandin species in, this is uterine tissue sections at different days of pregnancy. But it's very challenging with the isomers. So the isomeric structures that come from PDH2 are plenty. So all of these ones that I've circled in red. But the ones in squares, we don't expect to find because they're very short-lived. But that still leaves us with 3 isomers that are just different by very small amounts. But they're super important, probably, in the difference in the biological setting. So again, we turn to silver adaptation and tandem mass spectrometry. Here in MS3, we can see that PGE2 generates this diagnostic, more or less diagnostic product ion at 331. And delta 12 PGD2 has a diagnostic product ion at 341. Great, then we know who they are. But what about PGD2? No diagnostic product ions. All it shows us is that it's losing silver hydrate. Okay, and so does the other 2. They're also using silver hydrate. So now we had to take a different approach with our modeling, computational modeling. Again, we did the mixtures of the different ones, and we had to then instead do a multivariate regression modeling. But again, you can see here with the predicted and the true that we managed to get this with pretty good root mean square error here. So that means that we could then spatially find these ions. So this is, it says MS3, but this is actually MS1 image, so before fragmentation. Here are the MS3 images of then the 331, which was the DPI, diagnostic product ion, the 333, which was not, then the 341 that was. And if we then use our mathematical model to try to figure out where these are located, this is what it looks like. And sorry for the blue is kind of dark there, but they're all localized at this AM site of the uterine tissue here. This is where the uterine tissue will sort of die off eventually to have the embryo move into, what to say, grow in that direction. So that will be a part. And the other part, the one that's called M here, is where the uterine will form, or the placenta will form, sorry. Yeah, so we can see that they're all localized in this AM pole here, but a little bit a different ratio, as we can see. So prostaglandins are I actually asked ChatGPT this, and this is the reply. Why are prostaglandins important? I got all of this. So they're important in a lot of things. But the last story I want to tell you is about cholesterol homeostasis in the brain. And cholesterol is very difficult in cholesterol isomers. There are a lot of steroid isomers. So we all know this, but this is actually the first time someone has been able to image the distribution of steroids in the brain. This is a PhD student in my group. And we can see that they have quite different distributions. Some of them are not so abundant, like the estetrol here, but some of them we can see quite interestingly. So this is all done in MS1. So that means that we're again summing up and we are assuming that these are the steroids that we're seeing. But if we then use silver adaptation, and here we were using ion mobility, for those of you who are mass spec people, this is a cyclic IMS system. But in ion mobility, what happens before the mass spectrometry detection, we can separate the ions based on their drift time. So basically based on their 3D structure. And we were interested in seeing if we could differentiate between pregnenolone and 20-alpha, dihydroprogesterone. And when we're then, this is one pass of the cyclic IMS. And so when we're looking at the protonated ion, we see pregnenolone and 20 dihydroprogesterone here. They're not separating. So on the x-axis here, we have drift time in milliseconds. So that is how long it takes for them to go through this drift tube. But they're not separating, so it's the same. Similar with the sodiated adduct, similar. But when we have the silver adduct, we see a separation. So that suggests that they're changing their conformations, the 3D structure, when they become silver adducts. So that was quite interesting. So then we decided to simulate what are their structures here. So we used a bunch of programs for this, all available online or for everyone, so to speak. And what we could see was that when we're looking at the sodium adducts, they tend to sit in the double bonded oxygen molecule. This is where the structure is most energetically favorable. So that's why these sit here. But with the silver, it's a little bit different. So we can see for the 20-alpha dihydroprogesterone, the silver also sits on the double bonded oxygen there. But in the pregnenolone, it actually forms a complexation with the oxygen and with the double bond. So that results that the pregnenolone actually has a boat conformation in this part of its molecule, instead of the elongated conformation that we see in the hydroxyprogesterone one. So that is what's behind this differentiation in the ion mobility structures. And we, of course, looked both theoretically and experimentally on this, and it's within 5% or so that these collisional cross-sections that were calculated are. What we could also see is how do they fragment. So this is again the driftogram here, and these are different isomers. So as you can see, they are more similar here. So they're just different by where the hydroxyl group is. So this is hydroxyprogesterone isomers. And they do separate here in the drift time. I don't remember how many passes this was. It might have been 6. But anyhow, what we can see when we're looking then at the fragmentation pattern of these silver adducts, we can see diagnostic product ions for all of them. And so we continue to do that for lots of different standards that we have for steroids, including here we have corticosterone and dioxycortisol. And we can see that by charge remote fragmentation, because silver sits in one place of the molecule, and when it induces energy, it will fragment at a different place. So that's called charge remote fragmentation. Then we get some specific silver adducts. And after looking at all of our data, we realized that these are specific to the D ring and the E side chain. So that means that we have some diagnostic product ions formed in the D ring and E side chain based on the silver adduction and tandem mass spectrometry, which is quite exciting because this is where a lot of the isomerization happen. So this is a very important site. So when we're then looking at imaging here in MS, so the top row here is MS1 and the second row is MS2. So we can see that the first two are kind of similar, a little bit different in intensity, but still. But if you look at the androstenediol, you see that there are actually differences in the MS1 and MS2, which suggests that the MS1 has other molecules below it. And this is even more clear when we're looking at a precursor ion that has 2 different isomers under it that are then in the lateral ventricles here, only specifically compared to if you were looking at the whole time. So we wanted to take this a bit further and look at the dysregulation of steroids in disease. So we know that it's neurodegeneration, diabetes, and other ones that are important. And a collaborator of mine is interested in multiple sclerosis. And cholesterol is being a major part of myelin. Of course, we were interested in if cholesterol oxidation would have some impact in multiple sclerosis and demyelination. So when we're using white matter brain tissue from non-neurological controls and people with MS, what we detected were all of these 4 cholesterol and 6 cholesterol products here in the brain tissue. And if we look at the distribution, so on the left we have 3 controls, and on the right we have 3 MS tissue. And we can see that we're detecting them particularly in specific regions here. By looking at it this way, it's really hard to say anything about anything, right? So what we did was we combined this with their myelin staining, PLP. And so this was done in Gijs Kooij's lab in Amsterdam at UMC. And they stained then the PLP to see where do we have myelin and where do we not have myelin. And based on this, and so we had different tissue sections, so they are consecutive here because of the staining protocols. So when we then compared with ours, so what we can derive from this comparison is that there are different regions in the tissue that have different amounts of myelin. So we have the normal PLP, the diffuse PLP, and the absent PLP regions here marked as the different colors of blue in these. So then we could transfer this information to our tissue and extract data just from these regions of interest that we are interested in, and looking at the role of oxysterols and cholesterol in multiple sclerosis. So if we first start looking at cholesterol, so that's up here to the, there. We see that cholesterol decreases as demyelination increases. So in demyelinated regions, we have less cholesterol, quite significantly. If we look to the right, we have the hydroxycholestonic acid, and here there is a trend of increase in the demyelination, but it's not quite significant. The p-value is 0.053. So we have very low number also, as you saw. So there is a trend, but D-dihydroxycholesterol and homodeoxycholic acid, they actually are significantly increasing in the demyelinated regions. So that suggests that they are important in these regions of multiple sclerosis. And if we are then converting the data a little bit to look at oxidation level, so that is how highly oxidized are the species that we find in these different regions, we see there is an increasing trend of cholesterol oxidation in the increasing demyelination. And to the very left there, we also see the controls in the yellow and the MS, multiple sclerosis, in the orange. And we can see a huge difference in the oxidative environment with the oxidized cholesterol here, if we're just grouping them by that. Yeah, so in summary, I've shown you that we're using silver to enhance sensitivity, but also selectivity in both ion mobility and in tandem mass spectrometry, or MSN, by fragmentation. We can detect silver isomers and see what they are quite specifically. We use diagnostic product ions for phosphatidylcholines and steroids, and also mathematical modeling for fatty acids and prostaglandins that didn't have the specific fragmentation patterns. We can find the positional prostaglandins, the double positional fatty acids, prostaglandin regions, and steroids with this method, which is quite exciting, I think, by using just nanodeci or pneumatically assisted nanodeci together with silver and a high-resolution mass spectrometer with tandem mass spectrometry possibilities. And also here I showed you the first results here. We just submitted the manuscript on this cholesterol oxidation and multiple sclerosis. So it's quite excited about this. And so I want to, of course, acknowledge the people who did the work. And here we're actually in the silver mine in Sala in Sweden. So I thought it was appropriate for this talk to put the picture in here. Everyone's not in there, though, as you can see. But we have Kyle and Leonidas, who worked on the prostaglandin projects, Johan, who did the phosphatidylcholine and also the fatty acids, Varun has been on the steroids and the oxidized cholesterols, and Gabor on the multiple sclerosis, and of course our collaborator for the multiple sclerosis and Amsterdam UMC. And funding from various funding sources, otherwise it would not have been possible. And also I want to take the opportunity to let you know that I have one postdoc position and one PhD position announced on the Uppsala University job page. If you are interested, please take a look. And thank you very much for listening, and I'm happy to take any questions.
Thank you, Ingela. That was a fantastic talk. There's already a lot of questions. Let's go in the back on row 6. Yeah. Is there a microphone?
Me now? Yeah. Okay. Hello, Artem Pavlovsky, CBMR. Thank you very much for your presentation. I have just 2 short questions. First, are there any analogs of your technology in, let's say, market? I don't know if you're selling this or just want to know if there are analogies. And second, could you also mention some other collaborations you have? Because I feel like this technology can go far beyond biology. For example, in criminology, you can test trace of substances on surface, right? Maybe. So I'm really interested if there are any examples of who you worked with.
Yes, okay, great, thank you. So I work with quite a different crowd of people that are interested in biology or biomedical things. So we have been doing something with a little bit of diabetes. We've been looking at anastomosis. Everything is not published yet, so it's in a way. So generally what I find that I do is that I go to communities and talk and figure out what is the lacking thing, and then we publish this, and then people usually reach out to say, oh, can we do this together? So we're always open to collaborate. I don't have specific examples for you right now, but it's a very good point, and I meant to say it in the beginning, that this technique that we're using is not commercially available. There are commercially available techniques to do spatial metabolomics. The reason why I like this one is because it's modular. So if we look at MALDI or DESI or TOF-SIMS, all of these, they are ionizing at the same time as they're sampling. So what I like with NanoDESI is that we have the separation of the sampling event and the ionization event. And that allows us to play more with what we can do with it. So this is why I stick with this. But if you want to do other techniques, it's of course also available commercially. This is not, yeah. Then there's an issue about, sorry, yeah, we can deal with that later, yeah.
Take a question here.
Hi.
Jose Flores from Mass General in Boston. Very nice talk, and thank you. Very clear, so easy to understand, and thank you for pioneering methods that benefit the whole community. The question I have, you've shown how with your sliding capillary, you can obtain serial measurements of metabolomic samples across an anatomical space. And the question I have is whether you might be able to use parallel sampling with multiple capillaries at the same time, say, for example, after a perturbation, so you can have a real-time progression, in parallel.
If I have parallel mass spectrometers, that would work nicely. But the issue is
with a lot of machines.
yeah. So I think I really like the idea. I assume that you would have to save the sample though, because right now we're just directly spraying it and then it's gone, right? But maybe there's a workaround.
Paul says, it'd be nice to see a time course across multiple regions at the same time after you change the environmental conditions of the slice, for example.
So we're using now frozen sections, so there are no conditions here, they're not alive. But yes, absolutely, I'll be happy to discuss that because I think that's quite exciting, just monitoring the shifts in the metabolome over time. I think that's a very interesting approach.
Hi. Salvatore Rizza from Danish Cancer Institute. Thank you, amazing talk, as I say, and thank you for developing this method that we need. So we are working on mitochondria, and we are doing some lipidomic analysis, trying to identify changes in cardiolipins, and we have some good data. The problem is to identify the specific subtype and isotype. So have you tried to study cardiolipins, because they have 4 fatty acid chains, so the complexity is even more than phospholipids.
Yes, yes, exactly. So we see diacylglycerols with the 2. We see the triglycerols sometimes, but we don't really see the cardiolipins as far as I know. I haven't specifically looked for them either, but I think the ionization efficiency is quite low because they're quite heavy, right? But I don't know. It could be possible to target them. We haven't done that yet, but it could be a very interesting molecular group to target. Yeah. Yeah.
Hey, thanks for a very exciting talk. And I'm not an MS expert, so please excuse my ignorance. What is the limitation in resolution in this? Could this eventually be used to, for example, look at phospholipids of endosomes within a cell, different populations? And another question, when you prepare those slices, would you expect that differences in buffers during fixation, for example, would affect then the metabolites that you can measure in the end?
Very, very good questions. So we don't fix. So we're just using fresh frozen tissue. So when the tissue is extracted from wherever it used to be, it's just flash frozen directly, and then we cryosection it, put it on a microscope glass slide, and monitor it. So I don't expect us, except for the time course that we may have enzymes still available and potentially any oxygen. We haven't seen that, but then otherwise I don't expect us to change anything actually. And the first question was?
Regarding resolution?
Yes, resolution, yes. So this is not a high resolution technique, what I described now. It's more of a cellular region type technique. There are high resolution techniques. And if you want to do subcellular, there is those that work with second ion mass spectrometry. So TOF-SIMS and NanoSIMS that can actually look in an individual cell to look at those fine structures. They have the caveat that they have very high energy as they're hitting the cell, so then they're fragmenting quite a lot. So with nanosims, you only see elemental compositions. But if you're smart about your experiment and have isotopes in for what you want to look at, then you can still look at it. But they're not seeing intact molecules as we do with the Lecter spray so much. So their solution here, the best that my group has published is, I think, 20 times 50 micrometers for 1 pixel. So that's a few cells. My postdoc advisor in the US, Yulia Laskin, where this technique was developed, they have made a microfluidic chip. And with this chip, they also get something around 10, 20 microns, a little bit better that they are using, but still not down to single cell perhaps, but not quite to subcellular. And I think it's just a physical thing that you have to have the liquid bridge there. But I would go for other techniques if you're interested in a subcellular. Yeah.
Thanks.
I think there was one question more here. Final question, short, please.
Thank you so much. My question could be quite naive for a chemist, but so when you're ionizing the metabolites, how do you know that the adduct is on a very particular place? Do you predict it, or you also test it?
Yeah.
If you test it, do you test it for the whole metabolome that you detect in your analysis? So, I mean, do you verify them one by one?
So generally, we don't really care where the adduct sits. So for us, most often, we're just interested in seeing the ion, and then we're super happy. But there are some, I mean, of course, the adduct will sit at the most stable part of the molecule. So you can sort of understand approximately where the adduct would sit, and also based on the fragmentation pattern, you would understand. So for this ion mobility thing that we did, then we were interested because it was such a big difference when it was silver compared to the other ones. So then we did simulations, and that can be done, of course, for everything. But generally, we don't really go that far because it doesn't really matter so much for us.
So the mass spectrometry itself tells you the story.
Yeah.
Thank you so much.
Mm-hmm.
All right, please join me thanking Ingela again for a wonderful talk.
Thank you.
Thank you.